It's my pleasure to present the work we are doing as the Sampling and Sample Processing Committee, SSP. I will present only one part of our work, which is the work we are doing on species selection processes for ERGA and BGE, or we have done. So first of all, let me introduce you to, sorry, just 1 minute. To the SSB committee. We have a coordinator, Rita, then we have several steering committee members, but we also have a lot of people who are contributing also to the work we are doing, especially what I present to you now. We're doing very different stuff, and if you are interested in joining us, please do that at sample at or contact us as at sample at erga-biodiversity.eu. So today I will present you about the species and species prioritization process and why do we need such processes. The first, and why should we select species? We have all these large genome consortia, but even with these, there's usually more species to be sequenced than funding allows. So we need in some form select the species we want. We need to develop criteria and strategies on how to select the species. This is done in different ways, and in ERGA BGE, we choose to use a bottom-up approach to involve the community in it. And with this, we mainly meant then the SSB committee, but also the ERGA Council. I come to this in a bit. So how did we proceed in developing these strategies? First, we started and compiled and assessed the individual selection or individual selection criteria. We defined them, we looked at the possible impact. Based on these discussions, we decided on the relevant ones. And these relevant ones we grouped into different stages and categories. And so we came up for the whole selection process on 4 stages. The first stage is the exclusion, which we had 3 criteria in there. Then we had the stage of prioritization, where we have 11 criteria in 6 categories. And the third stage is the feasibility check. And then the final stage is that all permits are in place, so that the species, the samples have been legally obtained. So with respect then to the prioritization, we looked a little bit further and we developed different ranking procedures, which we call models. So the first thing in doing this, we determined of the 6 categories we had, which were the most important ones to us and which were not so important to us as a representation of the community. So we looked at this, and for example, what you can see here is that the taxonomic representation was clearly favored by everybody, whereas, for example, applicability was not so much favored. So based on these information we know on the importance, we then move forward. But to show you that with taxonomic representation, this is not just a gut feeling. If we look, for example, as of this end of July this year, you can see the representation of reference or complete genomes per phylum. We see many genomes, many phyla, have very little representation of these. Many have none or only one genome represented, whereas only very few have a very strong representation, as we can see here, with more than 100 species. So next, with these importance for the different categories in mind, we developed different ranking models, in total 8. Within each model, we have the same weight for each category. So the weights within the category were not changed. For example, here is model 7, where we used a decision tree, where in the first level we decided was based on taxonomic representation. Then on the second level, we have country representation together with certainty of species identification. And the third level, we have different JEDI criteria. And on the fourth level, we have the applicability and novel leader as the ones which were then important. We had then these 8 different models, and these we tested using simulated and empirical data to assess, and we also looked if they produced the desired effect we wanted, which we said which were important to us. And to assess this, we used the factor relative enrichment factor, by which we measured how much a category got enriched into the top selected species in comparison to its contribution to all species. And this was set relative to the maximum possible change. In total, we generated for each data set 1,000 species, and then we generated almost 73,000 simulated data sets to test different combinations of factors, probabilities, and all of this. We did the same for empirical data, where we also looked at enrichment, applying the same factor, but we also looked there at the intersection between the selected species, how many of the models selected the same species or not. Then we presented these processes and results to the ERGA Council, which has at least one representative from each European country, and this should give us a good sense about what the community all over Europe thinks about the species selection process. And then in the end, the Council voted on what process should be preferred, and it was the Model 7, which I presented you before, was clearly favored. And we implemented additional ranking spec favoring both countries and individual researchers. And this was done after the Model 7 was applied, but before the feasibility check. So here is now the whole process. It's now outlined for the BGE community sequencing. So we have a set of graded species suggestions. This goes into the exclusion stage. If one of the exclusion criteria is not fulfilled, then the species suggestion is excluded. We go to the second stage, to the prioritization stage, where we first apply the Model 7 with taxonomic representation at the top level. Then we go to the further ranking, where we keep the top 10 ranked species unchanged. Then we move up the best ranked species for each country, which is not among the top 10 ranked species. The next step is to then look at the individual researchers, move up the best ranked species for each individual researcher who's not among these above groups. Sorry, that was above ones. And then we rank the remaining species in accordance with the decision tree. This goes then into the feasibility check, and all species not fulfilling the feasibility criteria are excluded at this stage. Then we have the last final ranking. We had several genera for which there was multiple species suggestion. And given that taxonomic representation was so important for us, we kept only the best one and moved the others to the bottom of the list. With that, we had a final list of species, and from there we counted from the top the first 38 species because they added up to the total genome size of 50 GB, which is what we can provide in the first round of the community sampling. We also generated a list of species on a waiting list, which we have 14 species, which added up to 25 GB. So we get different lists now where we can actually look into and work with this list of selected species, species on a waiting list, non-selected species, non-feasible species, and excluded species. All the sample providers were informed about the decision, and now they're sampling and sending the sequencing to the sequencing centers. And at the fourth stage, now we actually before the shipment, there's a check that all samples have been really legally acquired. If that's not the case, we cannot sequence them. This is 38 selected species after stage 3, but what I want to now look into a little bit more is like what effect had the selection process. Originally, there were 22 filers suggested, and 11 out of these were among the 38 selected ones, which you can see on the left side. Interestingly, of these 11 excluded ones, 10 out of them were due to the feasibility check, not due to the prioritization process. When we look at the country, because that was another important part by the ERGA Council, that it's evenly distributed across the country, 16 out of the 25 suggested countries were considered. Exclusion, again, was mostly due to the feasibility check. So from the other countries, they suggested species which were not feasible. At the individual level, we have a better representation. We have only one researcher, which got to provide 2 species. In all other cases, it was 1 species, 1 researcher. So kind of what the council really wanted. At the end, now I wanted to look at the effect of the feasibility check. So half of the species, which were suggested, almost half the species, were considered not feasible. That's quite an amount of species which are not feasible. Why was it so? They were too small in body size or cell numbers, or too large in genome size. 28% were too small, 18% too large. So in total, roughly 45% fulfilled at least one or both of these criteria. The other thing which stick out was they were in too bad conditions. About 23% could not snap frozen upon sampling, which is not good for DNA extraction afterwards. 37% could not be preserved within 5 minutes after their death, and 28% it was not possible to maintain a strict cold chain. So all of these are not beneficial for later DNA extraction and good quality DNA in the later process. The third category was that they are too challenging to collect. 18% of the species were not already collected or were not easy to obtain and were due to this excluded. So what it shows us that feasibility actually, the feasibility check on non-feasibility is a large proportion. So in conclusion, for my talk now, the species selection process we developed was a bottom-up process with community involvement. It's a 4-stage process with a total of 28 objective criteria. It's an automated selection process based on R scripts. There's no human intervention in the selection process itself. And what it clearly shows, we need more research effort to make the sequencing of reference genomes feasible for a larger part of the biodiversity community. With this, I stop sharing.